you giving your time today to the Research Interest Group's April webinar. We have today with us a distinguished guest from CHOP, and I will just, before we get started, remind you of some things coming up on our calendar. Our all-member webinar is scheduled for this time 2 weeks from today, and it's focusing on this year's Action Day. It's making our voices effective, getting ready for Action Day, and you'll be being sent a reminder about registration for that if you haven't done so already. The meeting team would also like to remind you that the link to register for our annual meeting, which will be there in Philadelphia at the Children's Hospital of Philadelphia, is available, and we would like you to get registered. Elise and Lisa Towery are still collecting information about funded research for the ICRP database. If you have not submitted that yet, they are waiting for you anxiously. We also would like to remind those folks who are going to be in Washington, D.C. in May for Action Days that there is you can register and also get hotel reservations now. I would also like to call your attention. I think that the value slide is up next. Is that right?
Yes, it is.
One of the things that we always do before we have any of our meetings or webinars is to remind people of what brings us together and makes us effective, and the most important of which is always to put children and their families first in everything we do, as well as do these other, we share these values to support each other and to be accountable and cost effective and inclusive and really to stay action oriented and flexible. Today we have several folks that are CAC2 members involved in this webinar, including Amy Weinstein, who is our technologist and moderator. She is the executive director of a Kids Brain Tumor Cure Foundation, also known as PLGA. We also have with us Donna Ludwinski of Solving Kids Cancer and Julie Sutherland of Make Some Noise Pediatric Cancer Foundation, and they will be helping Amy triage your questions, because I really do believe that they have the expectations that you will want to make this an interactive webinar and take advantage of having our distinguished guests. You are muted right now and will remain muted. I believe that it will work better to triage these questions by using your control panel to pose the questions that you have. I'm now going to turn this over to Julie Sutherland, because she has important updates and information about the Research Interest Group and CAC2's planned research conference for the fall.
Thank you, Vicki, and welcome everyone. So we are, I'm sure most of you know that we have a research conference coming up and in the fall at Cold Spring Harbor Labs. It is unfortunately over Halloween, but we're going to make it fun, October 30th through November 2nd. This is a first ever meeting of its kind where it's a collaborative meeting where we're bringing everybody, all the stakeholders involved in childhood cancer to this meeting to share ideas and learn more about the current research going on, what next steps, and all the information is on the CAC2 website. If you go on the landing page, there will be a slide there after the summit meeting. You can go in the research section or you can go in the event section. You can find it there. When you get there, you will find information about sponsorships that, you know, if you'd like to share it with any funders you might know, if you have any relationships with pharmacy companies or anything like that, you feel free to take that packet and share it with them. Research members are also encouraged to sponsor in whatever small amount they would like to support. Everybody who is a CAC2 research member is going to have their logo on a step and repeat banner at the conference. So we're all going to be represented, and our community will be visible to everyone. But feel free to also send in some sort of contribution in the sponsorship of the meeting. The registration link is not up yet. We've been waiting on Cold Spring Harbor's. They're having a little issue customizing our page for us. but it should be up by the end of the week. So check back next week to the website and see if the registration link is active. As we gain sponsorship money, there's a very good chance that we will be able to offer partial travel scholarships to CAC2 research members, because we want to be sure that everybody can make it to the meeting. And I guess that's it for now. Vicki, did I leave anything out?
Sorry, I was muted. No, I think that you did cover everything that needed to be covered, and I thank everybody for this, and I hope you enjoy the presentation.
Great. Okay. So, Julie, are you all set then?
Yes.
So my name is Amy. Okay, great. So this is Amy Weinstein. Welcome, everybody. I hope everybody can hear us well. I'm really excited to be the moderator on this webinar. This is a really unique opportunity for our organization, CAC2, to come together. And we are incredibly fortunate to have with us today Dr. John Maris, who is a pediatric oncologist at the Children's Hospital Philadelphia and the co-head of the Pediatric Cancer Dream Team. He holds the Giulio D'Angio Chair in Neuroblastoma Research. And as you all are aware, he is going to be speaking today about the topic of epigenetics, pharmacogenetics, and pharmacogenomics. I'm hoping he's going to explain to me the difference between those two things. But more importantly, he's going to be talking about the process of learning causation in pediatric cancers, mutations, and really critically finding targets in childhood cancer. So we're going to ask Dr. Maris to begin shortly, but I just want to say one quick thing, which is that this is really meant to be, as Vicki mentioned earlier, an interactive webinar. We are counting on you guys to submit your questions via the control panel, which you should be seeing on your screen. There's a question link there. Just type your question in. And Donna Ludwinski is going to be helping us at the end of Dr. Maris's presentation, and she will be helping triage the questions, and we'll get a good flow going with that for the last 20 minutes. So without further ado, I'm going to turn the reins over to Dr. Maris. Dr. Maris, welcome.
Well, thank you very much, and it really is my pleasure to be here today. I want to thank Amy and Julie and Donna for the invitation, and hopefully you're seeing my screen and you can hear me well. Can someone confirm that?
You're good, John. Thanks.
Okay, good. So you're an ambitious group of people for sure, and the topic that I was asked to speak to is really broad. Epigenetics, pharmacogenetics, pharmacogenomics, and the title that's on there. I tried to keep this more focused on the latter part of it, and I'm going to try to go through this slide deck rather quickly, famous last words, and to leave plenty of time for questions. But I really wanted to try to focus on this is more of a personalized medicine sort of talk. There's a lot of buzz of personalized medicine, and I wanted to talk about what it is and what it's not. Before I dive in, I just wanted to tell you one anecdote that I think this group would find of interest. Is in the roles that I have, I had the opportunity to sit with the vice president 2 days ago to discuss pediatric cancer, and I was quite, I've always been a Biden guy, and I was really impressed by his knowledge of the pediatric cancer space and how prepared he was for the meeting and the questions he asked and what he knew and what great questions he had. So I really think that you as advocates should really be pushing on this moonshot thing to make sure that the pediatric cancers are represented. And if we have time at the end, I can talk about what I mean by that, because it all ties into what we're going to be talking about here today. So this is a loose outline. I know that all of you are very sophisticated in your understanding of childhood cancers, but I know that there's a broad level of knowledge about the science behind it. So I want to sort of make the assumption that you have not thought about science since high school or college, and just make sure we're all on the same page with some definitions, and then talk about what the promise of personalized medicine is, the opportunities and the obstacles in a very pediatric specific way, and somewhat of a vision of the future. And again, I study neuroblastoma, but I've tried to make this broader than neuroblastoma, but many of the examples will be with the disease neuroblastoma, but I think that it largely represents all of the childhood cancers. So you've heard of personalized medicine, and you know what that term means is an individualization of patient care. And while many people think that this is something new, it's actually been around for a long time. And you know, what we're really talking about is personalized genetic medicine, and that is using this ability to understand what's right and what's wrong about our DNA, and to be able to have the technology to get those readouts pretty quickly, and to apply that both for prevention, precision diagnosis, and treatment of disease. And then it gets a little confusing because some people talk about personalized medicine and precision medicine, but they're the same thing, so don't think that they're something different. The field of genetics, and again, the terminology can be very confusing, which is why I wanted to go through this. Genetics is really the study of the genes that you were born with. And then you hear people talk about genomics, and it's really cancer genomics, and this is the study of how these genes become disorganized in cancer cells. And I think that many of you remember, or probably many of you know very deeply, that the rule of life is that all of our information for what makes us look the way we are and act the way we do and get the diseases that we get is encoded in our DNA, and then that gets translated into RNA, which then goes to protein, which is the target that we're trying to hit with our drugs, whether they're small molecules or immunotherapies. So, and obviously this DNA to RNA to protein is modified very dramatically by the so-called epigenome. And I'm not going to talk about any of that specifically today, but would be happy to answer questions. Hitting the target and proving that in a patient is the field of pharmacogenomics. And again, I'm not going to get into that in any detail today for time limitations. And then all of you have heard about DNA sequencing. It's also often gets referred to as next generation sequencing or next gen or NGS. So you'll hear these terms, and all this is just this amazing technology to read through the 3 billion letters that organize our information of life, and to define what is right and picking the needle out of the haystack for what is wrong. So again, I apologize for those where this may seem way too basic, but I wanted to make sure we're on the same page. The word mutation gets used a lot, and it is a very simple term. It's any alteration in the normal DNA sequence. But I wanted to make sure that you all understood that it's not at all clear what is normal. Everyone's sitting in their various computer screens have different DNA. There are so-called polymorphisms which make us as human beings different. So it is often somewhat challenging to know exactly what is the reference or the normal, but there is a consensus reference human sequence, And mutations are simply spelling mistakes or breaks in the chromosomes that bring chromosomes, the strands of life, together in an abnormal fashion, or mutations that cause too many copies of the gene. Normally you have 2 copies of every gene in your body, one from mommy and one from daddy. So many cancer mutations cause dozens or hundreds of
Dr. Maris, I think we've lost your audio. Dr. Maris? Julie, can you hear Dr. Maris?
Yeah, I'm
more and more tumors.
Yep, there we go. Okay.
Was that a question for me?
Actually, I lost your, I did lose, I don't know if other people did as well. This is Amy Weinstein. I did lose the audio on you. You were talking about me. Nope, go further forward.
Okay. Can you hear me now?
Yep.
Yes.
And I got through some of the mutation slides.
Okay.
What is normal you were talking about? That was the last I heard. So maybe, would you mind
well, that's too bad.
I apologize. Okay.
Well, let me go back then. So, and I'll try to do it quickly because I don't want to run out of time here, but a big issue in understanding what's wrong in DNA is understanding what's right in DNA, and everyone on this call has subtle differences in their DNA, and that's what makes us unique as human beings. So these are so-called polymorphisms, or how we've evolved to have different eye color and different hair color. But there is a consensus about what is normal, and then mutations are any mistakes in that, and these can be changes in the lettering of the DNA, breaks in the chromosomes that bring them together abnormally, or we normally have 2 copies of each gene, one from mom, one from dad, and cancer cells can figure out ways to amplify this to have dozens or hundreds of copies of genes, or to delete one or both copies, and these are all different types of mutations. And then also, very importantly, mutations can either be germline or somatic. Germline means that they're in every cell of your body, they were inherited, or they can be inherited, or that they're restricted to the cancer cells only. And these are very important differences to think of as we're gaining information at an individual patient level. I'm not going to get into this in any detail, but there's also this important issue of mosaicism and heterogeneity, which means that that some mutations occur during fetal development, so that it's not in every cell in the body, but in a subset of the cells in the body. That's mosaicism. And that I'll talk about the heterogeneity issue a little bit later, because this is also very important. Okay, so just a cartoon of what a mutation looks like. Our DNA is double-stranded, and it's in this beautiful helix. And the top just shows, a spelling mistake, and how you can have a mutation just by there being one DNA letter that is mistaken. And then the bottom in this stick diagram is a picture of a protein that's very important in neuroblastoma called ALK. And the stick figure at the top is a cartoon of the protein that we're targeting with drugs. And the bottom stick figure is a blow-up of the so-called kinase domain. And this is how these different mutations are reported. So if a patient has a spelling mistake in the 1,245th amino acid, which happens to be an F, or so-called phenylalanine, that would get reported out as a mutation. And this cartoon is showing all of the known mutations in the alpha kinase gene. So again, I just wanted to show some examples of what we're talking about when we're talking about mutation. So I think you're probably all aware of the vision of personalized cancer care. It's to precisely identify patients predisposed to develop cancer. While this may not seem like such an important issue for kids, it really is, because we're learning, as I'll point out, that a larger subset of children than we expected likely have a bad gene in all their bodies that they could pass on to their future children and or lead to the potential for cancer later in life, a second cancer. There's a bullet in this, and we want to use this to provide much more precise monitoring for those who are predisposed to a first or second cancer, to provide much more precise prognostication for children newly diagnosed with disease. Where I think most of the attention is right now is to develop much more rational and precise therapy. And we'll talk about that a little bit. And then we're developing very rapidly techniques to monitor mutations in the bloodstream through these so-called liquid biopsies. And this is a very rapidly moving field to try to get a genetic proof that a patient's in remission or early detection of a relapse. And then finally, as I mentioned, we really want to use this information to help tailor the actual doses and schedules. Everyone metabolizes drugs differently, and we're learning how to predict that based upon tumors and blood DNA sequencing. And the point of this slide is that while all of the genomic stuff is new, it's not all that new. So we have examples. We've been caring for children who are born with something called Li-Fraumeni syndrome, which is a mutation in the p53 gene, for years. We've used MYC-N amplification in neuroblastoma since the 1980s to help with prognostication. We've recently, in the last 5 to 10 years, made some precision advances in ALL and neuroblastoma. The leukemia field has figured out how to do molecular monitoring of leukemia for a long time. And there are examples of how we adjust drug doses, some very common like gentamicin, others like this so-called TPMT and leukemia that you change the dose of 6-MP. So pediatric oncologists have been leading the way in this, but I think that there's been a great acceleration over the last few years, which is why there's been so much attention played. So this is a very complicated slide that I took from a recent publication from a group at the University of Michigan. And what this is, the point of this is, what does tumor sequencing do for you? And listed at the bottom there are a bunch of different diseases, as you see, and then across the top, these are just a bunch of different genes that we have in all of our bodies. These are genes that happen to be known to be important in cancer, and they work through different ways. And then if there's a color coding at the bottom, there's some sort of alteration. So these were about 100 patients who came to the clinic in the University of Michigan, and they all had their DNA, they all had their tumor and blood DNA sequenced to see if it could lead to a change in therapy, or could help the patient. And all of these things are things that one day we should be able to do something about. Now, this manuscript was both very important in showing that you can learn a lot, but it was also very sad in some ways because very few patients could benefit from the information learned. And most of the time it was because by the time they got the data back, the child was too sick, or because they were not able to access the drug or drugs that they thought might be able to help a patient based upon a mutation in one of these genes here. The other very important point, and I think you saw the arrows fill in here, is that many of the so-called actionable issues were unsuspected mutations in the germline DNA. So that these were patients who did not have a family history, but had mutations that they may have inherited from one of their parents or arose new in them, and they could pass on to future children, and may impact second cancers if they're survivors. And so this was a very important paper, and I list over here on the right 3 other papers came out soon after this that had very, very similar findings. I also wanted to emphasize this paper that received a lot of press that came out of St. Jude, which was trying to quantify the frequency of germline mutations in childhood cancers. And with these plots at the bottom, and I'm not going to go through it in any detail, the bottom line is that 8.5% of the children in their study, and it was a very large study of over 1,000 children, had a mutation in one of these genes that are known to predispose to cancer. And so, for example, the breast cancer gene, very well known to lead to breast and ovarian cancer, but was not known to contribute to the variety of different cancers that happened in these children. Now, we're still in an area where we're learning, what does this mean? Did the breast cancer gene actually cause the rhabdomyosarcoma in this one patient, or did it just happen to be there and had nothing to do with the rhabdomyosarcoma, but is important for knowing for future risk of breast cancer or ovarian cancer? And so there's a huge amount of momentum in this field, and while we've learned a lot, there's still a lot that we need to learn, and I hope that that was clear. I'm going to skip over the bar graph in the top right, but I did want to make sure that one point was very, very important, is that I was taught when I was going through medical school and training that that if you did not get a family history of cancer, then it was very unlikely that the child had a mutation in a cancer predisposition gene. And what these words at the bottom basically say is that's not true. That we've learned that many of these mutations arise in the sperm or in the egg, so the parent isn't affected, but the mutation is new in that patient. And so it's not so much that they inherited the disease, but they could pass it on, and as I've said a few times, could be at risk for other malignancies later in life. So to sum up this portion of the talk, cancer predisposition is very complicated, and I think I've emphasized that. We've learned a lot, but we still have a lot to learn. As we'll get into in just a moment, the cancers themselves are even more complex and more dynamic, and I'll show you some examples of that. And then another major obstacle to our field is that the vast majority of childhood cancers, you don't need a biopsy to diagnose relapse. And so how we've learned a lot, but we have not been able to study the most important samples to many families and the whole community, which is the samples after they've been exposed to chemotherapy, radiation therapy, and I'll show you why I make that point, why that's important. So this is another complicated slide that I just want to try to walk through, and let me just come back here, I'm sorry. So this is a study that actually my group published where we looked at neuroblastoma. We had 23 consecutive children where we had their tumor at diagnosis, and then these children all received chemotherapy and then suffered a relapse, and we had their tumor at relapse and were able to sequence them. And what these wheels show are all of the chromosomes in our body sort of organized this way. So this is chromosome 1, this is chromosome 2, and then these little letters out here, hopefully you can see my pointer, are genes that were mutated at diagnosis. And then the green are chromosomal areas that were lost, and red were gained. And then this is the relapse sample, and I think you can see that there's just a whole lot more going on. And if you look at this bar graph, what this is showing is that, for example, in this patient NB1269, what this is saying is that there were about 100 mutations in this child's tumor at diagnosis, but about 250 at the time of relapse. But only about 45 of those were shared. And so the very, very important point here is that if a child is at relapse and we're trying to assign that patient to a new personalized medicine therapy that may take advantage of mutations in the tumor, if you send the old specimen from diagnosis, it doesn't reflect what you have when the child goes on that trial. And that's why I think it's just such a very important point for the community to try to understand why we're going to be more and more insistent on trying to access tissue at the time that patients are going on clinical trials. So in my opinion, it used to be, is the risk of a biopsy too high? And I think that we have learned enough, and there is the potential for benefit so that we are in equipoise. And I can tell you at my institution, our interventional radiologists have gotten very, very good at performing these biopsies as outpatient procedures with we think, very little risk to the patient. And so, obviously different when you're talking about a brain tumor, but there are brain tumors that can be biopsied safely, and so this is probably an area that we'll have time to have some discussion. And then there's a lot of hope in this liquid biopsy that we can get enough DNA from the circulation so that we don't have to go into tissues, and that's really important for many cancers that are in difficult to access places. So the last sort of broad area that I want to cover is, so you do all the sequencing and you get a readout of different mutations. But it's very important to realize that the presence of a mutation does not mean that we have a known effective therapy. First of all, mutations in different diseases often behave very differently. So a BRAF mutation in an adult melanoma behaves very differently than a BRAF mutation in a pontine glioma or a low-grade astrocytoma. A drug that can kill a melanoma cell based on inhibiting BRAF in melanoma will actually activate and accelerate a low-grade glioma. And then even with that complexity, I showed you before all the different mutations in ALK. Some of them are very easy to inhibit with currently available ALK inhibitors, and some are completely resistant. So this is a major issue, and where the pediatric oncology community is really struggling right now is that there are just not enough investigators who are doing the critical experiments to define which of these DNA mutations with potential drug matches actually makes a difference. Which of these should be studied in the clinic. And this is an example of how it should be done. So this is from Yael Mosé, who's down the hall from me, and she's been studying crizotinib in this disease, neuroblastoma, and this is just showing the amount, the degree with you're able to inhibit the activated kinase. So the more the bar goes down from the open bar, the better it works. And this is what I was trying to say on the prior slide, that this mutation is easier to inhibit than this mutation and this mutation. So in patients, we've seen these patients respond. These patients who have these mutations don't respond. but she was able to obtain a new drug that looks great across them all, and then she took this into mouse studies, and these are the exact sort of data that you want to see before you open a clinical trial, where the tumor in the mouse grows very quickly. So this is weeks. This is the size of the tumor in the mouse. It grows very quickly. With the ALK inhibitor we used to have available, crizotinib, it slows things down, but if this was a clinical trial and you did an MRI right here, that would be progression of disease, right? So this wouldn't be a failure. But this new drug, regresses and just cures these mice. And so you can see multiple examples of the old drug not doing so well, the new drug doing much better. And these are the exact sort of data that we just need to have across all of the spectrum of different diseases and different types of mutations. So I think that, as a community, we have to be very careful. There are a lot of clinical trials that are opening up that are borrowing from the adult experience, but there isn't the right preclinical data in pediatric cancer models. And I think that we just have to be very, very careful. And those of you who are advocating for families presently or in the future, we need to query, what are the data other than just the mutation. And there's also, a lot of things out there that are sound too good to be true in terms of quote-unquote personalized medicine trials. And if they sound too good to be true, they often are. So, for example, there are a few clinical trials where they say they'll take the tumor out of the patient, put it into a series of tests and test tubes, and determine what drugs work and what drugs don't. But that completely ignores the fact that the tumor changes completely once it comes out of the environment of the body. And these so-called mouse avatar experiments are doing the same thing, but in a mouse model. And this holds some promise, but none of this has been validated yet. And I'd be happy to answer questions. So I think it's very important, and I just don't have time today to get into this, but we could do it in the question. Immunotherapy is another form of precision medicine. It's defining the right target and defining the right drug, whether it's a CAR T cell or an antibody or an antibody drug conjugate. And there's a huge amount of progress in the field in the childhood cancers and defining better immunotherapeutic targets than what we have. In neuroblastoma, which I study, GD2 is a good target, but it's not a great target. And there are many others that we're trying to push forward very quickly. And then I already made this point, and I forget why I had it in here again, is that we are just at the very beginning of understanding the genetic basis of toxicity that we see. So, precision medicine with having, the best ALK inhibitor, that's not just going to replace chemotherapy, radiation therapy right away. Maybe not ever. There's going to be an integration. And we are getting much, much better predicting, for example, what children are more at risk for cisplatin-induced hearing loss, doxorubicin-induced heart failure, other things. And this will be just a part of precision medicine, and this is the so-called pharmacogenomics that we didn't get into in any detail. I think that this is my last slide. The future is, in the near future, there's an urgent need to credential personalized approaches. I have neuroblastoma here because this was from a prior talk, but all pediatric cancers. So we've done a lot, but at this point on this day in 2016, we have not proven that all the sequencing and stuff is worth it. It's expensive. It's I'm a believer. I think it is going to be proven, but we have to prove that it's all worth it, that we can improve patients' lives. We have anecdotes. Everybody who does this has anecdotes, but we need to prove it in a broader way. And as I already said, and for those of you who are involved in fundraising and directing research dollars in this space of translation from the genetics to a clinical trial, there just has to be a dramatic investment. I mean, I think we need exponentially increase the funding in that space, encourage investigators to get into that space. It's hard to compete for NIH dollars in this space because it's often derided as very applied or very, it's not hypothesis driven, but it has to get done. And I think that's really important. So with that, I'd be happy to ask any questions.
Dr. Maris, thank you so much. That was quite succinct and very layperson friendly. So I, for one, without a science background, really appreciated that. This is Amy Weinstein. I'm gonna turn the webinar over to Donna Lunwinski, who is gonna help us filter through some of the questions. But I also just want, before I did that, I want to just acknowledge and thank you for really being direct about this issue of raising funds. I find that there are a lot of physicians and a lot of clinicians and researchers who aren't quite there yet, making that connection, and I think having the clinicians and the researchers advocate that private groups, public charities need to raise funds is really critical to increasing the groundswell. So thank you so much for acknowledging that. Donna, do you want to get started with the questions?
Sure. Thank you, Dr. Maris. Our first question is from Julie, and she wants to know what the community can do to be helpful or proactive in getting these secondary biopsies at relapse as a standard procedure. Do you feel like that hurdle has been, surmounted, or do you think there's something our community can do about that?
So I think it's like anything that is a culture change. It's going to take time and it's going to take perseverance. No, it's not a done deal. And it's as much on the side of the oncologist as it is the families. I have not met a family where when the subject of a second biopsy has come up that they have, assuming that they understand the reason for it, that they have not wanted to do it. But I have met many of my colleagues who have not really bought into that this is worth it. So I think that this is more on the pediatric oncology medical community than the parents. So I think that it's, if you are advocating for a child or a family where the word is that, well, the doc doesn't think a biopsy should be done, I certainly would want to understand that a lot more and or consider a second opinion.
Very good. You did a nice job talking about the obstacles and pitfalls, and one question that often comes up in the community is, should there be a targeted mutation and a drug that might seem appropriate, and there is no trial available, so the families and the physicians seek compassionate access. Do you see that as a major obstacle right now in the field? And also just the burden of time that it takes for the teams to access these drugs. What's your opinion on that situation?
Yeah, thanks, Donna. It's a huge issue, and I could probably just take the rest of the time talking about that issue, but I'll try not to. There's a huge problem with access. So I'll try to make a couple of points, and they're not necessarily in any logical order. First, in terms of advocacy, it's my opinion that there is a significant amount of risk aversion that we need to get around. We're talking about children with lethal diseases. Often when we're seeking these, there's little or no hope of a cure. Yet, most pharmaceutical companies are very reluctant to provide these drugs. And it used to be that the FDA was also very risk adverse, but Rick Pazder has changed that around very significantly. So it used to be that the FDA was as much of an obstacle as the pharmaceutical companies. But now it's the pharmaceutical companies, and you know, I'm not blaming them, and I'm not saying that there's not a reason to have some risk aversion. You know, their nightmare scenario is that a drug that they've invested billions of dollars in, literally, there'll be some terrible side effect in a kid that wasn't anticipated. But that's an unrealistic fear, and it's just hard to convince them. I think that Nancy Goodman has been working pretty hard to address this issue, and as many of you may know, just announced the opening of through their Kids First program, that they've developed a suite, a web-based suite to help physicians get compassionate access. According to Nancy, in a conversation we had recently, this is, you know, to help them get the IND, to help them with all the regulatory stuff. The huge burden that is placed upon the doctor and the nurse when they're doing this literally can take days of work to get this done. We'll have to see if that's helpful, but I'm hopeful it is. So it is a huge issue. The other major point I want to get is that what we really need to see over the next few years is more and more innovative clinical trials that are set up so that we don't have to go get compassionate access for a newly discovered mutation. So there needs to be an effort in the field. You know, we have defined all the mutations in the last few years. We know what the mutations are, so we know what we're dealing with, and we need to begin to design the prospective clinical trials so that children who could potentially benefit could get onto these trials and have the drug provided within the context of the clinical trial, not only to benefit the patient, but also to help us learn where we are helping and where we're not.
Oh, that's a perfect segue to the next question. Talking about getting investigational agents in trials, and you and Dr. Mose have been working for a while on the Nepenthe trial. Can you tell us a little bit about the impact of actually having a trial that is going to potentially include combinations of investigational agents?
Well, what Donna is referring to is a clinical trial for children with neuroblastoma that is called Nepenthe. It stands for Next Generation Personalized Neuroblastoma Therapy, and it is starts at the time of relapse with a biopsy. The tumor gets sequenced, and then, unlike other trials where there's some molecular tumor board or consensus, and then you select therapies, the protocol specifies if you have mutation X, then you get therapy Y. And very important in our mind of how to do this trial is that we wanted the therapy to always be combination therapies. And so, as Donna said, this took a long time to get organized. It is now, you know, after a year and a half or 2 years of working pretty hard on it, fully FDA approved, and is at our local IRB to be reviewed next week. So we hope to be enrolling patients within a month or so. But it was complicated. But we went through this with the hope that this could be a template for similar clinical trials in other diseases, other institutions. We're opening this at CHOP, but that we very much hope to expand it to many other institutions once we get it up and running. It was just impossible to tackle the multi-site part of this right out of the gate. And you know, we hope that it's a kind of trial that would help credential this sort of approach and prove that it's worth it, but we will have to see.
Well, that's fantastic. It's very exciting development, because as you said, just the impact of designing a trial in that fashion is really exciting. Going back to the germline mutations, you had mentioned the St. Jude's paper that had quite a few tumors tested, and it only showed 8.5% that had a known cancer predisposition mutation. Do you know if those are continuing to be discovered, or is that a pretty firm set of mutations? I've heard that there was suspicions that there might be more predisposition mutations that are discovered over time, but I'm just curious about the status of that understanding.
Yeah, the paper was pretty recent, so where we are in the field is it's not clear yet if the 8.5% number is an overestimate or just the tip of the iceberg and an underestimate. And I actually had the opportunity to write an editorial for that paper in the New England Journal, and I tried to explain why. So on the one hand, that study might be an overestimate because they happen to have many patients in their study with known cancer predisposition syndromes. Children who have a special form of ALL called hypodiploid ALL almost all have germline p53 mutations, for example. So some argue that it's an overestimate. I argued, however, that it's more likely an underestimate, and that's because they only looked at a very, very small percentage of the genes, and they had very strict definitions of what was a potential causal mutation. And so my prediction, which is based on more hunch than data at this point, is that as we continue this research, that number is likely going to go up. The other very, very important point is that it's not just as simple as one mutation predisposition. It's also possible that there are two less potent mutations that are interacting, and so I'm a believer that childhood cancers arise largely because of genetic influences. Not that any parent should feel blame because they passed it on. These are almost always newly acquired genetic changes that happen because mutations are always happening, and we usually repair them, but not always.
I see. Well, thank you. For one last question, since last year there was quite a lot of buzz about the NCI pediatric match trial that was under development or discussion. Can you give us an update on that and what you think the impact of that trial is likely to be?
I think that it's moving forward. It is, a high priority for both the Children's Oncology Group and the Center for Therapy Evaluation Program. And I don't know when it will open. I know they have a target to get it open in 2016. I won't be surprised if it's 2017. I have a feeling it's going to be a stepwise process. The biggest handicap this has is the point that I tried to really emphasize, that a lot of the proposed mutation drug matches in the trial have no preclinical data. Just taking a page out of, it worked in melanoma or it worked in colon cancer, hopefully it will work in kids. And I think that, there's going to be a little bit of catch-up to try to get preclinical data. So the hope for the match is that it will be a study that's around for some time, and I think it will build. And at a minimum, it will allow us to learn a lot more about relapsed childhood cancers. We still don't know how Ewing sarcomas evolve after chemotherapy, how osteosarcomas evolve, so we'll learn a lot. And you know, the key for the advocacy group is to help us get the best drugs into the study. You know, there are 8 or 9 ALK inhibitors out there, but there's one that I showed you as best in class, and that should be the one in the match study, not one of the others. And sometimes CTAP can get certain drugs but not others, and I think that that's going to be a really key part of the match study.
That's
Dr.
extremely
Maris.
helpful to know.
Yep.
Go ahead.
Dr. Maris and Donna, thank you so much for doing the Q&A portion of the webinar together. It's 12:57. We're going to have to, unfortunately, wrap up this webinar. But first, again, I want to just thank you, Dr. Maris, for sharing your time with us this morning. It's a pretty safe guess to say that the organizations and the individuals representing the organizations that are on this call share your enthusiasm for raising awareness and funds to really jumpstart some of these critical, especially innovative efforts that the government won't otherwise fund. And we really appreciate your sharing your perspective. So thank you so much for that. Julie and Donna, I just want to thank you for organizing the webinar, giving credit where credit's due. This research interest group would not happen without the efforts of people like Julie and Donna. So thank you both very much on behalf of CAC2 for organizing this. The next webinar, the research interest group webinar, will be in May. The details of that webinar will be forthcoming in Vicki's monthly email, which goes out at the beginning of May. I just want to remind webinar participants that this webinar was recorded, and we will have a recorded version up shortly. So if you've missed the beginning part, or if you missed the end, or you can feel free to share it with other people as well. So once again, thank you so much. I'm going to sign off on behalf of the research interest group at CAC2. Once again, thank you, Dr. Maris, and thank you to all of our participants.
Thank you very much.
Thank you.
Have a great
Thank
day.
you very much.